Hi, everyone. I'm Carolina Petrillo, a postdoc in Professor Hans Noakes' lab at Columbia University. And my talk today is focused on better understanding how mitochondrial dynamics regulate interferon and age-related changes in hematopoietic stem cells. As a background, we know that blood cells are predominantly short-lived and hematopoietic stem cells are required throughout life to replenish multi-lineage and more committed precursors. Our lab is particularly interested in the role of mitochondria in hematopoietic stem cell biology, since even if these cells rely on glycolysis and not on mitochondria for energy production, they have actually higher mitochondrial mass than lineage-committed progenitors and mature cells, so suggesting an unknown roles of mitochondria in hematopoietic stem cell biology. And mitochondrial roles in cellular health are several, not related to ATP production for cellular energy, but actually they are also involved in other cell mechanisms such as gene expression regulation and also growth production, release of apoptotic factors, communication with other organelles like the ERs, endoplasmic reticulum, and also a role in innate immune responses. And actually, the lab reported a very important role of mitochondria in HIC maintenance. And in particular, in this paper, they found that mitofusin 2, called MSN2, which is a protein involved in the fusion dynamic process, has a crucial role in the maintenance of HIC with extensive lymphoid potential. And they also identified the mechanism of action, which is dependent on calcium and NFAT signaling. And this is actually a really important aspect involved in HIC aging, because with age, hematopoietic stem progenitor cells expand. They become more myeloid biased with a concomitant lymphoid defect. And these lead to loss of self-renewal and regenerative capacities upon transplantation, and also deregulation of the innate immune system and inflammatory responses. And for this reason, I want to better investigate its role in the biology and also during hematopoietic stem cell aging. And to directly address the role of mitofusin 2 in hematopoietic stem cells, we used wild type and mice with conditional deletion of MSN2 in the hematopoietic cells, driven by the valve-Cree system, since the global knockout is embryonic lethal. And using EDU DNA incorporating reagent, we found a significant increase of HSC proliferation in the absence of MSN2, maintained in both fraction with myeloid and lymphoid potential. And this is also in line with a significant increase in the frequency of hematopoietic stem cells and multipotent progenitors in the bone marrow and also in the spleen of MSN2 deleted mice, suggesting an expansion of hematopoietic stem progenitor cells in medullary and also extramedullary compartment, and a role of MSN2 in maintaining overall HIC quiescence. To understand the mechanism behind this phenotype, we performed RNA-seq on wild type and MSN2 knockout hematopoietic stem cells combined with single-cell RNA-seq on bone marrow and spleen, revealing a striking upregulation of the type 1 interferon signaling pathway, and in particular of interferon-stimulated genes, also called ISGs, but not the interferon ligand itself in hematopoietic stem cells, as well as in bone marrow and spleen from MSN2 knockout mice compared to the wild type. And this is like just an example representing IP27 expression, which is an ISG. ISG inserted hematopoietic stem cells, so which the expression is higher in the knockout cell population. We also validated the upregulation of few interferon-stimulated genes in sorted hematopoietic stem cells and also in differentiated lineage-positive cells from wild-type and knockout mice. So overall, it seems that the interferon signaling induces hematopoietic stem cell cycling in line with literature. Moreover, detectable interferon alpha but not beta was also found in the serum of MFN2 knockout mice, but not intracellularly from RNA-seq. And interestingly, deleting STAT1 or the type 1 interferon receptor together with MFN2 fully rescued the effect of MFN2 deletion on interferon production, hIC cycling, and also hIC frequency in the bone marrow. And this data suggests that MFN2 must be a negative regulator of the type 1 interferon signaling in hematopoietic stem cells in a STAT1 and interferon receptor dependent manner. Of course, now the question remains where this interferon is produced. And since aging of hematopoietic stem and progenitor cells is associated with the stem cell expansion, as well as a deregulated inflammatory responses, we analyzed the role of MFN2 during aging. And we noticed that deletion of MFN2 accelerated the aging phenotype in young mice with relative expansion of the stem and progenitor cell compartment in both medullary and extramedullary organs, which is more similar actually to the old mice with exacerbated loss of lymphoid potential and myeloid skewing during aging. And moreover, performing a competitive bone marrow transplantation in lethally radiated recipient mice, we showed that MFN2 deleted young hematopoietic stem cells sorted from the bone marrow and from the spleen have actually less reconstitution capacity. Also in this case, more similar to the old hematopoietic stem cells, confirming a stem cell impairment. So this proves that MFN2 deletion leads to an increased number of phenotypically defined hematopoietic stem cells with less self-renewal and regenerative capacity, which are actually very important aspects of hematopoietic stem cell aging. And finally, we also showed that age MFN2 deleted whole bone marrow has decreased hematopoietic regenerative function, as well as skewing toward myeloid differentiation and lymphoid impairment with a full rescue deleting STAT1 together with MFN2. So we can prevent the aging phenotype accelerated by the MFN2 deletion, inhibiting the interferon pathway. And actually, the last question we have tried to address is where this interferon is produced, because we cannot find it in any hematopoietic stem and progenitor cell population in the bone marrow or in the spleen of MFN2 deleted mice. And for this reason, since the intestinal and lung microbiomes are a local source of chronic interferon signaling that is important because it maintains the immune alertness, we analyze, and these are really preliminary but promising experiments, mice treated with a cocktail of antibiotics for 2 weeks to eliminate the microbiota. And we actually observed an abrogation of the MFN2 phenotype, analyzing the hematopoietic stem and progenitor cell frequency, and also quantifying some interferon-stimulated genes in the bone marrow of MFN2 deleted mice versus the wild type. So to conclude, our hypothesis is that a key function of MFN2 is to shield hematopoiesis, and in particular hematopoietic stem cells, from chronic type 1 interferon production and signaling induced by the microbiome. And it is known from literature actually that the intestinal microbiome, for example, stimulate the innate immune system and hematopoiesis in a STAT1 and STIM-dependent fashion. So they both produce a type 1 interferon, which then can directly or indirectly bind hematopoietic stem cells producing interferon-stimulated genes. And in this model, we propose actually MFN2 inhibit or regulate tonic interferon signaling with players that still need to be discovered. And this will therefore provide important insight, of course, into the biology of hematopoietic stem cells, into the role of inflammation in the aging process, and also into the propensity of hematopoietic stem cells to give rise to age-associated hematological malignancies. And I want to finally thank my mentor, Professor Hansnow, and all the lab members for some support. My founding, the Cancer Research Institute, Columbia and Harvard Core Genomics Centers, the organizers to give me the great opportunity to talk today, sharing like my data and my project, and of course all of you for the attention. And questions are more than welcome. Thank you.